So what do we find? These are the numbers of skin lesions that we saw in 2011. You can see they're kind of scattered around the shelf here a little bit, but really concentrated up along the northern tier north of the Deepwater Horizon. And this is the same data for 2012. This hopelessly busy graph also includes, number one, the surface expression of the Deepwater Horizon. All those little yellow dots, though, are all the extant oil wells in the Gulf of Mexico. So you can see that not only do you have an overlap between the Deepwater Horizon and the oil wells, but the green dots here represent all the deep oil seeps. And so now we've got three alternative hypotheses that might explain why we have some skin lesions, if in fact they're toxicopathic skin lesions. So of course the neat thing is the West Florida Shelf has no oil wells. It has really no significant amount of oil seeps up on the shelf, and at least so far it didn't have any surface expression of the Deepwater Horizon, so that should actually be a pretty good control, right? Let me just skip to one of the bottom lines. For the northern Gulf of Mexico, what we did was we were able to replicate 26 of the stations. So this is the percent of the fish with skin lesions, and you can see that for a number of these species, the red represents 2011 and the gray 2012. Overall, the rate of skin lesions dropped by a factor of about 2.5, from a little over 2% to a little less than 1%. For some of the species groups, like snappers and groupers, it dropped by about a factor of 6, which is statistically significant. So bottom line, this was an episodic event and not some kind of chronic exposure, which kind of knocks out the theory that this is all based on natural seeps and historic things. So I think it's a pretty interesting story. One of the other things we found is that these are, I got everybody after breakfast, I guess. These are skin lesions. You can see there's a lot of scar tissue and healing up in these, and so what we found particularly in 2012 is a lot of healing lesions, which is a good thing. Of course, we can't document the animals that died, but that's another story. Let me skip that one. So this is sort of the other bunch of stuff we do, and that is drain the bile out of the gallbladders of fish. So when we're tearing apart the guts of these things, the gallbladders are kind of the key to understanding oil contamination, because of the liver is processing as if we all have a couple extra drinks on Friday night, your liver's working overtime to get rid of the toxic effects of that. Same thing with a fish exposed to oil. That's the gallbladder is the place to look for the metabolites of all that, and so we spend a lot of time looking at funky green fluid in the gallbladders. I wanted to point this one out. This actually is raising a bunch of ripples in oil circles these days. So these are different PAHs of polycyclic aromatic hydrocarbons. So this is naphthalene, and then these are the alkylated homologs of naphthalene, fluoranthene, phenanthrene, right? And so this is a sample that Chris Reddy took right at the wellhead when the oil was coming out. The red bars represent livers from 56 red snapper that we obtained from the northern Gulf of Mexico, and you can see that there's a really close, I wouldn't call a fingerprint, but an association between the relative composition of the oil coming out of the well and the oil in the livers of those fish. That's not a fingerprint. The fingerprint involves a lot more than that, but nevertheless, we can't reject the hypothesis that that's their oil. And the interesting thing, and the thing that puzzles so many people now, is you would think that there would be differential metabolism of these various PAHs because they all have different molecular weights, and so you wouldn't see this, that they would be differentially sort of metabolized by the animal as opposed to this. And so this has really got people scratching their heads quite a bit. I won't go through. This is a bunch of other things. Wanted to talk a little bit about some of the other studies we have ongoing. My newest student is Christina Deek, and Christina and I and Amy and some others worked together this summer out, collecting information from these fish. In particular, she's interested in other sublethal effects, and so she's collected a lot of blood serum and tissue, and she's going to be looking at immunosuppression as well as genomic changes through comet analysis and other things to look at these fish. One of the other studies we did this summer was for the NERDA people. And so we basically sampled these 9 field sites in coastal Louisiana. Terrebonne Bay sites over here are more or less a control. This is Barataria Bay. We saw some really nasty fish up in the northern part of the bay here. This is some sampling up there. Still oil up in the bay. Some really interesting fish like this alligator gar fish, which is a cross between 2 prehistoric, groups of animals. And then over here on the delta, these studies, we've got a number of samples from these studies, and these will probably be used in the case against BP. The last thing I wanted to mention was also as part of our lab, we're running the administrative side of the Sea Image Consortium, which is a large group of institutions, 13 different institutions. So with basically under the direction of Cheryl Gilbert, we're trying to coordinate what is an $11 million 3-year study with all of these partners. And we have, of course, we have a big meeting coming up at the Oil Spill Conference in New Orleans here in a couple of weeks. So with that, I appreciate your attention. Thanks.